So this presentation really is going to be presenting the initial findings of a multi-science analysis of human burials that we excavated from a Roman rural settlement in Somerset by Oxford Archaeology, in collaboration with a consortium of specialists, including the Archaeological Isotope and Peptide Research Laboratory at Durham University, the Francis Crick Institute, and Cardiff University Bioarchaeology. The project has been undertaken by Oxford Archaeology for Balfour Beatty on behalf of National Grid. Now, the work I'm presenting today, it's an ongoing project. The wider programme of post-excavation and publication work on the site has only recently started, but I'm going to be focusing on the headlines of our findings on the human burials to date, or some of the human burials. So the skeletons were found on one of several sites excavated by OA between 2019 and 2020 along a linear scheme called the Hinckley Connection Project, which involved the installation of electric cables between Bridgewater and Avonmouth. Now I hope you've got the title, the Jimi Hendrix link. Thank you. My husband didn't, so, and now he's going to watch this and be angry with me, sorry. So the works extended along 26 kilometres of the scheme north of Bridgewater and included overhead lines in the northern and southern sections and 8.5 kilometres of underground cabling in the central section through the Mendip Hills in an area of outstanding natural beauty. 6 areas, including 5 in the northern section and 1 in the southern section, were archaeologically investigated. And human burials dating from the Bronze Age through to the medieval period were excavated along the scheme. But the burials that are the focus of this presentation are from Roman contexts at the Maxmill Lane site that you can see the arrow is pointing to on this slide here. Now, Maxmill Lane is situated next to the Lochs Yeo River, within a landscape that is populated by a number of Romano-British sites, including villas, settlements, the Roman road to the north, shown by the hatched line, and the Roman lead mines and mining settlement at Charterhouse, just to the east, on the far right of the slide there. Previous excavations suggest a continuous band of settlement which extended from Wint Hill, shown just above Area 6, which extended down to the floodplain and to our site at Maxmill Lane. The site produced evidence for a large Roman nucleated settlement / small town with very deep stratigraphy and extensive archaeological remains dating from the late Iron Age through to the late Roman period. And this is just a selection of some of those finds and the Roman road in the top right there. We found 27 inhumations, including 2 prime adults, 1 possibly male and 1 possibly female, and 25 juveniles. The juveniles include 17 neonates, 5 infants, 1 young child aged between 1 and 5 years, an adolescent, and 1 that could not be aged more precisely than juvenile, that's less than 18 years of age. We've assigned them to 4 different phases. The earliest being Middle Iron Age or early Roman, and the latest being late Roman, but the majority are late Roman. Now, we have a programme of radiocarbon dating underway to refine this phasing, but for the purposes of this presentation, I'm just going to refer to them broadly as Roman here on. We're currently analysing the skeletons as part of a broader integrated study of the site, exploring themes relating to society, religion, economy, and population dynamics. Now, one of these themes, which is the focus of today's presentation, explores the relationship between mothers and infants. That is, what do the neonates and infants tell us about the maternal health, well-being, and funerary practices of the Maxmill Lane community? There are more detailed questions within this, but that's the broad scope. During intrauterine and early postnatal life, a child is entirely dependent on the mother for nutrients and antibodies via the placenta and lactation. As such, they aren't just a reflection of infant health and well-being, they are also a reflection of maternal health and well-being, even when the mothers have not been identified in the archaeological record, as at Maxmill Lane. They may provide information on, for example, maternal health/disease status, intra and extrauterine nutrition and social and cultural variables, such as responses to reproductive loss, and the funerary rites afforded to this age group. Our approach involves contextual analysis of the burials combined with 6 different types of scientific analysis, including osteology, amylogenin peptide analysis, DNA, histology, multi-isotope analysis, and trace element analysis. I'm a specialist in the first of those, so I'm not even a real scientist, but anyway, I'll carry on. These are being applied to bones and/or teeth from the adults and/or the juveniles. And we've coordinated this programme by making sure that we apply as many of these techniques to the same samples from each skeleton as far as possible, in order to minimise the impact of the destructive nature of the work and to maximise our results. For the isotope and trace element work, we've also analysed samples from 5 caprines, that's goats or sheep, and they've been selected to provide some baseline data. We estimated the ages of the juveniles using measurements of bones and/or by observing dental development. And as I've shown previously, most of them died at between 38 and 40 gestational weeks, referred to here as neonate, which is about the age of a full-term child. A mortality peak in full-term deaths such as this has been seen elsewhere and interpreted as a reflection of infanticide, a practice normally carried out after birth rather than stillbirths and natural deaths soon after birth, which result in a more dispersed age distribution. Now, while infanticide might be the case here, it's also possible that this mortality peak is a result a result of biases in the methods that we've employed to estimate the age at death from the bone measurements. That is, the methods have artificially inflated the number of neonates, when in fact the individuals reflect a natural mortality profile. And it's also possible that the bias towards neonates in this group reflects some sort of selection process which favoured them over other children for burial in this settlement context because of their age and other aspects of their social identity. One way of exploring these points is by the application of histological analysis to look at diagenetic changes in the bones of the neonates, as this may indicate whether they were still or live births. So in very simple terms, I did say I'm not a real scientist, experimental work has shown that the bones of a newborn infant that has had its first feed and subsequently died and was buried should have areas of bacterial bioerosion in their bones. A stillborn won't have a developed gut, so has no gut bacteria to spread around the body following death to cause this bioerosion. Let's have a look. So far, we have the results on the histology of 5 of the 17 neonates, and they all show evidence of bacterial attack, consistent with them all being live births, or in the case of one individual, a likely live birth. In the example that you can see here, the three skeletons, the bacterial attack is extensive. The appearance is very opaque, consistent with the babies having lived long enough in the gut microbiome to develop and so result in the bacterial attack that we can see here histologically. Spatial analysis shows that many of the burials cluster around domestic and agricultural buildings and/or structures. but there appears to be no distinction in terms of age or sex. Further contextual analysis of burial location in relation to domestic and non-domestic buildings, as well as specific features within them, might identify some patterns. So this is more work that we've got to do. So, for example, burial of newborn infants next to boundaries or in places of transformation, such as hearths or agricultural features, may refer to their liminal status, while burial in the domestic sphere may be symbolic of the connection between mother and infant. Sorry, that is what I was just talking about. Okay. Now, in a good number of skeletons, DNA preservation was good enough that the maternal lineage could be estimated. The initial screening data suggests that 7 of the neonates share the same maternal lineage, and they're highlighted, they're the red ones there on the end in that last column of that table. Interestingly, this maternal lineage has also been estimated from 3 Iron Age skeletons excavated from pit alignments approximately 3 kilometres southwest of Maxmill Lane at Webbington Farm, the site just down the way here. The similarity in the maternal lineages to each other is striking and so specific that it seems unlikely this has happened by chance. Full production data is now needed to confirm these initial findings, but it's interesting to note that while the maternal lineage is not itself unusual, the frequency of this lineage at these 2 sites is highly unusual to see, even at the initial screening stage. So what is this saying? Well, it could suggest that the individuals are either all close maternal relatives, or that they come from a society with low historic maternal diversity, or that burial at Maxmill Lane and Webbington Farm was partly informed by a matrilineal descent system. Further information on the mothers is reflected in the stable isotopes, which has explored their geographic origins from samples taken from the neonates and the infants. Now, geographic origins is usually explored by analysing oxygen and strontium isotope ratios in tooth enamel, but the neonates and infants were too young for this to be applied here. That is, they hadn't developed the required teeth for this type of analysis. So instead, we focused analysis on sulphur in rib samples from the juveniles and the fauna, the caprines. A relatively new method, sulphur can help to distinguish whether animals and humans were residing in wetland or dryland environments. And when analysed in juveniles this young, the values reflect the geographic origins of their mothers. In the case of Maxmill Lane, the results for 23 juveniles and 5 caprines show that the humans and animals both have mid-range values, suggesting that the animals were being reared close to or in a similar environment to the humans, and that they were living and sourcing their food on dry land, such as the uplands near the Mendip Hills, rather than in the Somerset levels. All the skeletons were examined for pathology, and most notably 4, including 3 infants and 1 neonate, had evidence of metabolic disease in the form of rickets resulting from deficiency in vitamin D and or scurvy caused by vitamin C deficiency, all diagnosed following published guidelines. In addition, 1 infant and 1 young child had the hair on end type of cribra orbitalia, a condition which can be the result of vitamin C and D deficiency, or it may reflect impaired absorption of iron resulting from these deficiencies. It's unusual to see this amount of metabolic disease in such a small group of very young children, and this could indicate that their mother's vitamin C and D status was also compromised. This suggestion that the pathology observed on the neonates and infants reflects compromised maternal health is supported by the reconstruction of their diets from bone collagen extracted from rib samples, because this shows that they had carbon and nitrogen isotopes, which were a trophic level above those of one max mil adult, which could be measured. That's skeleton 31790, shown on the slide there. So this confirms that most of the children were being breastfed. Interestingly, one of the infants, skeleton 39287, shown on the right there, had a high carbon isotope ratio, which suggests some input from marine or seafloor protein in their diet, or the mother's diet. So they and their mother might have been a migrant or a second generation migrant to Britain. Okay, last bit of science. Previous work by Joanna Moore and colleagues, shown here on this paper here, has demonstrated a direct link between lead poisoning and children with skeletal evidence for metabolic disease. It's well known that during the Roman period, populations were exposed to lead through the widespread use of heavy metal in everyday items, and in the case of Maxmill Lane, environmental pollution from mining activity at the nearby lead works at Charterhouse that I pointed out earlier on. Now, we haven't tested the Maxmill Lane neonates and infants for lead concentrations yet, but we have explored this in the tooth enamel of 2 adults and 1 adolescent, as well as 5 caprines from the site. And this shows that the caprines had high concentrations of lead, or off the scale, in to use Janet's words, Janet Montgomery. And this strongly suggests that they were grazing in an environment which was polluted by lead mining. In addition, the human tooth enamel shows elevated lead concentrations indicative of anthropogenic exposure to pollution. So, although this doesn't demonstrate a direct link between lead poisoning and disease in the Maxmill neonates and infants, it suggests that this could be a factor here. According to clinical research in these 2 papers on the bottom left here, maternal lead can cross the placenta and enter fetal blood circulation and can impair fetal growth. Further, human milk is a proven route of lead exposure for breastfeeding infants. Lead poisoning can disrupt normal metabolic function, leading to scurvy, rickets, and anemia, even when there's sufficient nutritional intake. As such, we suspect that the metabolic disease seen in the Maxmill infants is not the result of nutritional deficiency in their mothers, but is the result of maternal lead poisoning from environmental pollution. And we hope to test this by examining the enamel from the neonates for lead in the coming months. So that's a lot of information. Just to summarise and conclude, this presentation has described the scientific analysis of Roman inhumations from a settlement context in Somerset. In keeping with standard Roman funerary rites, most of the individuals were infants, and as seen elsewhere, most of these were individuals who had died at around the time of birth. Also in keeping with other studies is that the methods we've employed to estimate the age of these babies has probably artificially inflated the number of neonates in the assemblage, but only slightly. Histological analysis has so far found no evidence that the infants were stillborn, but more of the assemblage needs to be analysed before we can draw conclusions from this. A popular interpretation for these contexts is infanticide, but we're not convinced, and amylogenin peptide analysis shows that there is no sex bias in the group, suggesting that this is not an explanation here. In addition, there is so far no pattern in the spatial distribution of boys and girls in any age across the site. So perhaps age and sex were not factors in their burial location. The DNA evidence for genetic relations suggests a strong maternal influence on populations across the landscape, and perhaps this refers to a matrilineal descent system. Isotope evidence suggests mothers and infants were living and sourcing their food in areas such as the Mendip Hills rather than wetland areas such as the Somerset levels. Further evidence for metabolic disease in the infants, combined with isotope evidence which suggests that they were breastfeeding, highlights mothers whose health was compromised. Evidence for high lead concentrations in the fauna and the humans from the site suggests that this poor health was caused by lead pollution. This is a very powerful discovery that has great resonance today when environmental pollution is a global concern. So to conclude, contextual analysis of the infants buried in this Roman settlement in concert with biomolecular science allows a much more penetrating investigation of the population. It also provides a richer perspective on some old and new themes relating to the funerary treatment of infants, the living environment, fetal and infant health, and the health and well-being of their mothers, who are invisible in the archaeological record. This is a summary of the further work that we have left to do in the coming months. I also apologise because I scrambled my slides earlier and didn't properly present the peptide evidence for sex. Apologies to Jo there, because she worked hard to get that data for me. But do ask about that in the questions later on. And thank you very much for listening.